Welcome to the course Animal Diversity 1 Biology of Non-Chordates. Myself, Dr. Sneha, Assistant Professor, Department of Zoology, St. Joseph's College for Women, Vishakhapatnam. As a part of third topic from this unit, we are discussing about locomotion in Protozoa. And the content of this topic includes the various types of locomotory organelles present in Protozoa, the pseudopodia and its types, flagella, its structure and movement, cilia, its structure and movement, and comparison of all locomotory organelles of phylum Protozoa. After studying this topic, you will be able to identify different locomotory organelles such as pseudopodia, flagella, and cilia in protozoans. You can able to describe the structure and mechanism of each locomotory organelle. You can able to differentiate the various types of movements, that is like amoeboid movement, flagellar movement, ciliary movement with suitable examples. And you can able to analyze the role of locomotion in survival, feeding and habitat adaptation of protozoans. So let's talk about what is locomotion and what is the significance of locomotion in protozoans. Locomotion is the ability of an organism to move from one place to another place. So obviously in protozoans, though they are single cell animals, but still it is essential for them for searching of food, escaping from unfavorable conditions like extreme heat, too much overcrowded places, etc. like that, avoiding predators, and even for reproduction and dispersal also. Being a unicellular, protozoans use a highly specialized organelles for their movement. So in this slide, you can clearly see the various types of locomotory organelles what are present in various type of protozoans such as pseudopodia. Second type is flagella, and the third category of locomotory organelles are cilia. So let's talk about these organelles one after one. Let's start with pseudopodia. The movement using temporary cytoplasmic projections are called pseudopodia. Pseudopodia is also commonly known as false feet, as the name itself is indicating pseudo plus podia. Pseudo means basically false, podia means the feet, what they are having. Why we are calling pseudopodia as a false feet? Because they are temporary, they are not permanent in the type of protozoans actually. Okay, so whenever that particular protozoan it's wants to move from one place to another place, so during that time only they're going to form these cytoplasmic extinctions. So that is why for those extinctions they have named itself as pseudopodia, which means false feet. So mostly this type of pseudopodia locomotory organelles are seen in amoeba and that type of similar protozoans. So again, the pseudopodia are further divided into four different types basing on their shape and structure. So let's talk about those types of pseudopodia, that is lobopodia, which are actually looks like blunt finger-like projections, as you could clearly see here. So these are blunt finger-like projections what are projecting outside of the amoeba's body. So these are known as lobopodia, and the example for this category is amoeba. So the second type of pseudopodia is known as philopodia, where you can clearly see the thin filament-like thing what are projecting from its protozoan body. So this type of pseudopodia are known as philopodia, and the example for this is euglypha. Coming to third category of pseudopodia, that is what reticulopodia, where we can clearly see here, see the entire branching of network of cytoplasmic extinction. So this type of pseudopodia we're calling them as reticulopodia, and the examples for this category is foraminifera type of protozoans. Coming to last category of pseudopodia, that is what axopodia. where we can able to observe, see, the presence of stiff radiating axis like very sharp axis cytoplasmic projections are coming outside from the protozoan body. So this type of pseudopodia are known as actually axopodia, and the example for this category is radiolarians. So now let's talk about how these pseudopodia are going to be formed by the protozoans. So there are to explain this formation of pseudopodia, there are lots of theories were proposed by lots of scientists, but among all those theories, there is one theory which is much more accepted, widely accepted. That is what it is known as Sol gel theory of pseudopodial formation, which actually explains how these cytoplasmic extinctions are going to be formed by protozoans. Okay, so let's take an example of amoeba. So amoeba is known for formation of lobopodia type of pseudopodia, as you could clearly see in this diagram. So this diagram is actually clearly depicting or showing how in cytoplasmic extinction is going to be formed. Okay, so initially in the last topic we have talked about, as a part of general characteristics of phylum Protozoa, we have talked about in protozoans the cytoplasm is further divides into ectoplasm and endoplasm. Okay, so in this diagram you can clearly see the outer layer, which is much more clear in nature, the gel like thing, so that is what ectoplasm, and inner, which is much more denser, this area, this is known as endoplasm. Okay, so now see, suppose this amoeba, it wants to move in this direction, in this forward direction, like clearly see in this image. Okay, so what actually happens is, amoeba is an aquatic animal, so the water is actually starts enters into inside the body of amoeba, and because of this, what actually happens is the ectoplasm, which is a jelly like solution, it turns into very thin fluid like thing. Okay, and because the amoeba wants to move in this direction, so obviously because of entering of the water, what actually happens is it pushes the amoeba to form these cytoplasmic extensions towards in this direction. Okay, so later, once the amoeba moved in this particular direction, the water which is entering from in this direction, it exits outside from the amoeba's body. So this is how the ectoplasm and endoplasm, first of all, it turns the jelly-like ectoplasm and into that means the sol, which is basically a highly fluid-like thing, and later on, because the water is going to exit from the amoeba's body, so again that ectoplasm which actually gains it jelly like appearance. So this is what sol gel theory says actually, because of this solation and gelation process which is continuously happens. Okay, because of this process only the pseudopodias are going to be formed, and the protozoans, they can move in a forward direction or backward direction, upward and downward direction. Okay, so now moving to the second type of locomotory organelles, what we can able to observe in protozoans is flagella, which is something like more long, very long whip-like structure. Okay, so in this slide we're talking about structure of flagella. So this diagram is actually showing the transverse section or TS of flagella. Which is going to exhibit a very characteristic, very like the arrangement, very famous arrangement, that is what known as 9+2 arrangement, what we would say, because inside the flagella, if you're going to see, all these are nothing but the microtubules, a bunch of microtubules what are arranged. Okay, so now let's talk about this arrangement here. So now if you're going to see, towards that peripheral side, we can clearly see, so these are known as doublets, because in one bunch of the microtubules, they're arranged attached with two microtubules with each other. So this is what known as doublet. And if you're going to count all these doublets, they are basically comes as number of 9. On the other hand, towards the center, if you're going to see, so these are center microtubules which are present two microtubules separately. So these are known as singlets, which are actually 2 in number. Because of this arrangement only, this is known as 9 plus 2 arrangement. Okay. On the other hand, from each in this doublet also, if you're going to see, as I said that two microtubules are there. So in this, the front one is known as A microtubule and the second one is known as actually B microtubule. That is what makes a doublet actually. Okay. And from this B microtubule of each doublet, if you're going to see, a radial spoke-like thing is basically originating from each microtubule doublet actually. And these doublets, what are present 9 in number, they are also interconnected with each other. We clearly see here. So that interconnecting arm is known as actually nexin. Okay. And as well as the presence of these radial spokes like thing, radial spokes only. Okay. So they're going to provide the support internally and makes that flagella to maintain its structure as it is. So the best examples for flagellate protozoans like Euglena, Trypanosoma, etc. So now, after studying the structure of flagella, so let's talk about the types of flagella. Okay, so this flagella is again further divided into actually 5 various types like anematic, stichonematic, pentonematic, acronematic, and pentacronematic, basing on presence or absence of axonems. Okay, so here we would clearly see like presence of these small hair-like structures are present, and the filament-like structures also could clearly see at the tip actually. Okay, basing on presence of these two structures, the flagella is further divided into 5 different types actually. Okay, for an example, the first category, anematic type, could you clearly see it doesn't have that small hair-like structures, right? So that is why it doesn't have anything. It is simply looking like a simple hair-like structure. So this is what actually known as anematic type, completely that mastogonems are completely absent. Okay, now that is what, and the example for this category is cryptomonas. Okay, on the other hand, coming to second category, that is what stichonematic, where we can able to see actually the axonems are present only one single row. Okay, one single row, then that type of flagella is known as stichonematic, and the examples for this is astasia. Coming to third type, that is what actually pentonematic. Okay, so in this pentonematic, what actually happens, see, the multiple rows, that means see, the axonems are, sorry, the mastogonems are present on both the sides actually. Okay, and as well as we can clearly see here, okay, in this diagram, and the example for this is actually urocilus. Okay, moving to next, that is what acronematic. So in this acronematic, actually see, it doesn't have any mastogonems, but we can clearly able to see the presence of a terminal filament like this. Okay, now, right? So that is what a acronematic type, and the best example for this is Chlamydomonas. Coming to pentacronematic, if you're going to mix pentonematic and acronematic, that means actually presence of the mastogonems plus presence of that the naked filament. Okay, now, so it will become what actually pentacronematic, and the example for this is actually urocilus. Coming to the movement. So till now we have talked about types of flagella and structure of flagella. Okay. So now the flagellates, how they're going to move this flagella to move towards the front side, back side, or in the opposite directions. Okay, so basing on this, the different types of movements actually. Okay, so it was divided that flagella is going to move with the help of three different types of movements. Okay, so that is what known as paddle stroke, undulation motion, and as well as simple conical gyration. Okay, so let's talk about what is this paddle stroke. Okay, so in this, so this is what most common type of movement in flagella. Okay, where the flagella is going to move from one side to another side like this, right? So that is what known as paddle stroke. So in this paddle stroke, actually, we can able to observe two different type of stroke movement. That is what effective stroke and as well as recovery stroke. Okay, so during effective stroke, what actually happens, because of this effective stroke, the flagellate is going to basically push towards the forward side, right? So how this paddle stroke, sorry, how this effective stroke is going to be basically seen in this effective stroke, the flagella is going to become very rigid, like a stick, like very rigid structure. Okay, and it is going to move from here to in this by beating the water in this direction, right? So because of this beating, the flagellate is going to move towards the forward direction, and once the flagella is moved from here to here, right, as a part of effective stroke, so again it's supposed to go back like this, right? So that stroke where the flagella is basically moving from here to here, going back, as you could clearly see in this diagram, it is moving, going back like this, right? So that is what known as actually recovery stroke. Okay, so because of this effective stroke followed by recovery stroke, again one more effective stroke and recovery stroke. Okay, so that is how the flagellate is going to move in a frontward or forward direction. So that is what a paddle stroke, right? So moving to next type of flagellar movement, that is what undulation movement. Okay, so in this undulation movement, what how the flagella is going to move, the flagella is going to form a wave like pattern. Okay, right? So in this formation of wave like pattern also, the formation of wave happens in two different ways, like as you could clearly see in this diagram. The wave is basically traveling from here. See the arrow mark here. Okay, so the wave is traveling, the formation of the wave, it is starting from the base and it is ending at the tip, right? So because of this wave like formation where the wave is starting at the base and ending at the tip, it is going to create a pushing force where the flagellate is going to basically push towards the downside. It is basically pushed, right? On the other hand, exactly opposite type where the wave formation starts from the tip. Okay, here, see here. So from here tip to the base is formed towards the base. Okay, that is what tip to base movement of the wave formation. Because of this tip to base type of wave formation, it creates actually pulling force, right? So because of this pulling force, the flagellate is going to pull towards the front direction. So this is what actually undulating motion. Moving to third type of flagellar movement, that is what simple conical gyration. Okay, so what is this simple conical gyration is basically, see, as a part of this type of flagellar movement. Okay, so this is basically flagella, right? So the flagella is first of all turned into tightly like a screw. Okay, so once it is turned into like a screw or spring like thing, it is going to be released back suddenly, right? So because of this sudden releasing of the flagella, it is going to create a propelling force like this, right? As you could clearly see in this diagram, only this part of flagella is going to be moved, right? Which is basically turned first of all into screw, very tightly coiled like a screw or like a spring. It is going to be suddenly released actually. Because of the sudden release, what actually happens is it is going to create a propelling like motion like this, and the flagellate is going to basically move with sudden force. Okay, so that is what actually simple conical gyration type of movement. Okay, so these are the various types of flagellar movements that is what going to be exhibited by flagellates. Coming to third type of locomotory organelle, that is what cilia. So as you could clearly see in this diagram, the cilia basically, which are comparing to flagella, they are a little bit shorter, thinner in size, and they're much more number in size also. As you could clearly see it, this is what paramecium, the structure of paramecium, right? The the cilia is basically present all over around the body of the paramecium. Okay, so cilia are nothing but they are very numerous in number. They are short and small hair-like structures are known as cilia. Okay, the best example for this is all the ciliates and paramecium, as all of you know that, which is very well studied ciliate, right? And coming to structure of cilia, like they're almost all the structure of cilia internally is similar to the structure of flagella. Okay, so in flagella also we have talked about that 9+2 arrangement type of microtubules. So exactly similarly in cilia also we can able to observe that same type of 9+2 type of microtubular arrangement actually. Okay, so coming to ciliary movement, that means how the cilia are going to be moved. So similarly, like in flagella, how we have talked about, in cilia also, the effective stroke and recovery stroke is going to be performed as we have talked about. The cilia are going to turn into little bit stiff in nature, like here see, okay, and the cilia are going to beat like this, and the cilia are going to move in this direction. Okay, so the effective stroke is over. Again, the cilia are supposed to come back, as you could clearly see in this diagram. So that is what actually recovery stroke. Okay, so again performing the effective stroke and recovery stroke. So that is what one type of movement what we can observe in cilia. And one more type of ciliary movement is known as metachronal, like wave-like formation. Okay, where one after one the cilia is going to beat. Okay, so like see how wave is going to be passed. Like if you want to see any lush green the field, the rice field, actually see the wave is going to start from one side of the field and ends at the another side of the field. Okay, the beautiful wave-like formation we can able to observe. Exactly a rhythmic, basically the rhythmic movement of the cilia, one after one the cilia is going to beat upon each other, and they're going to form a smooth wave like formation. Okay, so this type of ciliary movement is known as metachronal type of wave formation. So this is what one more type of ciliary movement. Okay, so now let's compare all the different types of uh, the the types of locomotion what we have studied in this topic as a part of this. So we have talked about that protozoans are going to exhibit a highly specialized and that to three different types of locomotory organelles. That is what pseudopodia, flagella, and cilia. Okay, so now if you're going to compare these three in structure wise, as you know that pseudopodia are temporary formations, right? On the other hand, flagella is basically a very long whip-like structures, but they're very few in number. But on the other hand, cilia, if you're going to see, they are very short, tiny hair-like structures. But on the other hand, they are many in number actually, comparing to flagella. Okay, coming to speed. Okay, pseudopodial movement is considered as one of the slow type of movement, because for the formation of pseudopodia it's going to take lots of time, and even with that pseudopodia also they're going to move very slowly. Okay, on the other hand the flagella, moderate type of speed, but ciliates are one of the fastest protozoans what we're going to say, because of that beating speed of the cilia. Okay, and the type of movement if you're going to see, as pseudopodia where we're going to talk about cytoplasmic flow. Okay, now that solation and gelation happens basically, right? On the other hand, the flagella we have talked about by formation of that undulating type of waves. Okay, on the other hand, cilia are going to move with the help of beating actually, one after one, right? And the examples for pseudopodia, amoeba, the well known, and for flagella, euglena, and for cilia is paramecium. Okay, apart from this locomotion, these locomotory organelles actually, see for an example what type of additional role they are performing. So pseudopodia is not also going to act as locomotory organ, it also helps in feeding purpose also, catching the prey, like holding the prey, like that also. Okay, on the other hand, the flagella acts as a sensory type of organelles also, and even cilia is also going to act as feeding purpose for catching the prey, etc. Right? And apart from these three locomotory organelles, we can able to observe like some other type of movements also in very few protozoans. They doesn't have any specific organelles for this, but they're going to show this type of movement. Okay, for an example, like gliding movement, which is mostly seen in some type of specific parasitic protozoans. Okay, where, as I said, that they doesn't have any visible organelles. Okay, so they simply glide inside that vertebrate's body, like that. Okay, on the other hand, one more type of movement which is especially seen in euglena category, which is known as euglenoid movement, where as all of you aware of that euglena is externally covered by pellicle. Okay, so this pellicle is going to contract and relax, contract and relax. Okay, because of this movement of the pellicle, the euglena is going to basically move in a zigzag type of pattern, and this type of movement is known as actually euglenoid movement or metabolic, right? So that is what. So these are for this movement of euglenoid movement, gliding movement. They doesn't have any assigned organelles for that, but this type of movement is also seen, right? So, what is the significance of locomotion? Why they're supposed to need the locomotory organelles? Because it helps in their survival, adaptation. Because when they're moving to like new environments, actually from one place to another place, so they have to adapt to that type of environment. Okay, and definitely these locomotory organelles are essential for nutrition and feeding, right? And even they also helps in reproduction basically, because of that only they can disperse also from one place to another new place clearly. And especially in parasitic forms, like if they want to like that means transfer like from one host to another host for dispersal also, it is very very useful. Okay, so that is about the significance of locomotion and locomotory organelles. So as a whole, in terms of summary, protozoa move using specialized organelles, that is what pseudopodia, flagella, and cilia. So pseudopodia enables basically amoeboid movement, that is what through cytoplasmic extinctions, right? And flagella and cilia produces a rhythmic beating actually, as just now we have discussed that, right? And locomotion is definitely essential for survival, helping in capturing of the food, escape, and adaptation to the new habit. Okay, so these are the references are cited for this topic. Thank you for listening to the class.